Yeah, welcome back. This is another lecture which is connected with human genetics, and it is an introduction to pharmacogenomics. So first of all, you should know what is pharmacogenomics, and even before pharmacogenomics, the field of interest is pharmacogenetics. The pharmacogenetics is a study of variability in drug responses due to heredity. What we hold in our genes, because of that, we respond differently for a particular drug. So that is pharmacogenetics. And in our genes, this pharmacogenetics deals with one or two genes at a time. Pharmacogenome is what are the variation present in our entire genome, all the genes put together, how we are going to respond for a particular drug. That will be pharmacogenomics. And then there are so many factors which are involved in the drug response, and among that, genetic polymorphism is the one which deals with the pharmacogenomics. Apart from that, there are some environmental factors that also is influencing the drug response. They are age, gender, body weight, patient physiology, diet, smoking, alcohol, exercise, and drug interaction. So these are the environmental factors. How old are you, and how whether you are a kid or whether you are an adult or an old age person, depending on that, the drug may suit you or may not suit you. Depending on, say for example, you may have accumulated a lot of toxins over the years, and then certain drugs may react adversely to you, and then when you are young, it may be suitable for you, but as you are like 70-year-old, that same drug may not suit you sometimes because of its so these are the as a whole, the drug response and their variation is due to only these factors. Variation in drug response is due to drug interaction, environmental factors, and genetic factors. So in the genetic factors only is the study of pharmacogenomics style is only these three genes: drug target, metabolism, transport. Genes that are present in the human body does three different functions. First, it receives a drug. It is a drug target, and then it transports to the site of the action, or sometimes after the function is over, it is excreted outside the cell. So that is transporter protein which are involved in that. And metabolism, this is metabolic enzymes which are involved in converting a drug to an active form or some other form only. This will be functioning in or sometimes it may have to be once it is done, then the active form has to be deactivated before it is thrown away from the cell. So these are the genetic factors, and these are the other factors. Variation in drug response come because of genetic factors, drug interaction, and environmental factors. The earlier slide we saw all the environmental factors. Now we will move on to genetic factors. The study of pharmacogenomics and pharmacogenetics deals with only these three categories of factors. So drug metabolism. So the drug metabolizers are present in various parts of the body. They are nothing but enzymes. These enzymes are useful in converting the prodrug into active drug, or sometimes once it is done, then you have to, it may be toxic to the other cells, so you have to convert it to a inactive form so that it will be excreted outside the system. So hepatic microsomal enzymes which are present in the liver, and extra hepatic microsomal enzymes, they are present in the brain, and then in the heart and the other part of the body. And hepatic non-microsomal enzymes, all these metabolizers, the enzymes which belong to metabolizers will do these function: acetylation, sulfation, oxidation, reduction. They convert the drug into different forms so that it is either active or inactive in the system. Classical metabolism status. So some of the individuals are categorized as poor metabolizers, some are categorized as intermediate metabolizer, extensive metabolizer, and ultra rapid metabolizer, depending on what are the genes which are involved and what is the polymorphism present in that gene. That will categorize them as poor metabolizer. So for genotypes, homozygous null allele, heterozygous null and reduced metabolism, they belong to poor metabolizer. Homozygous reduced metabolism. So these are, in this category, you will have lot of genes, and the genes, whether they are present in the homozygous condition or heterozygous condition, and whether that enzymes are present abundantly or in a low amount, whether that genes are duplicated, all those things, genotypic changes will lead to the phenotypic change, which is called, categorized as poor metabolizer, intermediate metabolizer, like that. So depending on that, a particular drug, a poor metabolizer will not respond to the disease for the drug sometimes, because it cannot be metabolized. Extensive metabolizer, what he will do, he will easily metabolize it, and then even before the drug is available for the organ for correcting action, it will be excreted outside. So these are the high variations which is present in the two individuals. So the next category of proteins or genes which are involved in drug response is transporters. Transporters, drug entered the body, and only through transporters it is absorbed into blood, other organs, you can see here, transported to other organs and to blood also. So these transporters, if they are mutated, if they have some allele difference between the individuals, whether they will be transported properly or not transported properly to the organ of interest or for the excretion. So depending on the polymorphism, the drug response will be varying. So transporters can be classified into two: ABC transporters, ATP binding cassette transporters, and SLC, solute carrier. These are the two types of transporter proteins which are prevalent in the human. You nearly 49 genes classified into 7 families in the ABC category, and SLC 52 families with this many transporters. So they are active transporter, whereas solute carrier proteins are secondary. So what are the drugs like serotonin, dopamine? All these are transported by the solute carrier proteins, whereas this cystic fibrosis, transmembrane, P-glycoprotein, all these are different types of transporters. And then we will see the different type of targets. Targets is the first protein or protein which is come into contact with the drug, and then the drug is taken inside the cell. So the receptors, 45% of the receptors are drug receptors, and then some are enzymes, and some are hormones, and some are nucleic acid receptors. And these receptors are recognized, they recognize the drug and they make some changes, and based on that only this response will be varying. So now we have seen 3 different category of genes which are involved in drug response. So when they are showing some mutation, polymorphism, what happens in an individual? So what are the different changes that can happen in the individual? So as all are aware, individuals are having chromosomes, and then they have the number of autosomes and then sex chromosomes are given, and then they have lot of genes, and these genes which we saw earlier, transporters, metabolizers, and receptors, they also have the same structure of any other protein. So a single gene structure, eukaryotic gene structure, involves exons, introns. Exons are coding regions, introns are intervening sequences, and it will have promoters that are regulatory sequences. The polymorphism, the SNP can happen in promoter, SNP can happen in the exon, XNP can happen in the intron, or in the non-translated region at the 3' end. So if an SNP happens here in the exon coding region, it may change an amino acid, or sometimes it may not change the amino acid. If it changes the amino acid, the nature of the enzyme may be lost, or it may lose its structure and functions. That is possible. But what happens if the mutation happens in the intron? If it happens in the intron, sometimes these 2 exons may not be joined properly. So this particular protein may not be produced. So intron polymorphism is also equally important. Sometimes there may be mutation in the promoter region. What happens in the promoter region? If it happens in the promoter region, sometimes a polymerase cannot sit, and then it cannot produce the mRNA. So this protein will not be produced. Or if it is present in the promoter region, the amount of protein produced may be lesser. So it is affecting the expression of the expression of a gene can be. So for a drug response, one, you have to have a genotype. Another thing, you have to have a phenotype. Phenotype is what changes happening in the human being, that phenotype, or symptoms. Sometimes it is measured in symptoms, sometimes it is measured in when you treat a drug to a liver tissue, or you in vitro, people just liver cells, they take it and then treat the drug and then see what are the changes happening, how many genes are expressed and how many genes are not expressed, or how many genes are upregulated and how many genes are downregulated. That can, so that is the study of, say, pharmacogenomics in vitro, how people assess the drug responses. A test drug, a control drug. You can say test drug is say, for example, it is metformin. Control drug is nothing but water, you can say. So yeah, cell, if it is for diabetes, maybe some pancreatic cell or something like that. So this is also same cell, but it is just fed with water. When it is fed with the drug, what happens? What are the genes which are upregulated? So what are the enzymes which are triggered? What are the transporters which are triggered? What are the receptors which are triggered when the drug is applied? That will tell. So after applying a drug to a particular cell which of interest, what are the genes which are upregulated or downregulated? That will tell what are the transporter or receptor. We can identify those genes like that, and then based on that, you can also measure the phenotype. So toxicogenomics is another study. Pharmacogenomics is how the drug. Sometimes the drug may not be useful for our body, or it is already put into use, and now it has to be thrown away from the system because it has done its job, and what is the dose is high, so some of the things is not needed for the body, so the liver will toss it out. So when you look at the liver after administering the drug for some times, so then the liver decides, so this is too much for this particular individual, so this is sufficient for correcting action, and then rest it has to be thrown away. So what is present in the liver cells also will tell whether the drug has any adverse effect, because of these genes are triggered like that. So mutation, this already we have seen in a gene, and depending on what different region, the mutation will affect the drug response. So these mutations has to be, so the mutation only differentiate the individual. Some people behave differently for a drug because that individual is different because of the mutation present in those genes of drugs responsive genes. So that mutation could be either a single nucleotide polymorphism or an insertion deletion or a variable number of tandem repeat. Single nucleotide polymorphism is one nucleotide is varying. In the insertion and deletions, few nucleotides inserted in one case or deleted in some other case. Variable number of tandem repeats means some repeat sequences are present as 2 copies in one individual. Sometimes it may be few copies, or sometimes many copies is another individual. So those are the regions which show the genomic variation. Gene deletion and duplication. Sometimes a metabolizer is duplicated 2 or 3 times, means that person is going to make more of that enzyme, metabolizing enzyme. So this person will be an active metabolizer. His metabolism will be active, so he will be easily activating the drug, or sometimes easily he will be inactivating the drug also. So the phenotype has to be measured only by doing in vitro or in vivo analysis. So these are some terms which you have to know, and then how these are some of the technologies which are available for genotyping. Earlier lecture we saw eDNA barcoding. Similarly, for analyzing the variation in an individual, the gold standard is direct sequencing. So you have to amplify a region of a human being and then sequence it and find out what is the sequence, and that will tell whether he has any SNP or NDEL or variable number of repeat sequence. So earlier direct sequencing is the gold standard, but there are also other methods by which you can find out the single nucleotide polymorphism. One such is PCR RFLP, allele specific PCR. So for a particular allele, the PCR will work and then give an amplification. For a different allele, it will not give amplification. That is called allele specific PCR. And Tachman pyro sequencing. This is the PCR primer will be attached to a fluorescent probe. If there is an amplification, there will be a color detected. So this is as the it is the growth of the technology, and then later on high throughput sequencing. Now we have high throughput or hyper throughput Affymetrix Illumina. These are the chronologically the evolvement of the genotyping technology, but always completely sequencing that DNA. Directly sequencing is the gold standard. The DNA samples from human being can be either from whole blood, saliva, frozen tissue, plasma, or serum. So some of them are frozen tissue. Sometimes it is invasive procedure is needed to get the tissue. Say sometimes, but say saliva, this will have like dead tissue of the buccal cavities. So that is why we are using that. So DNA microarray is a technique which will help us to identify the single nucleotide polymorphism, which we call it as SNP microarray. When it is a cDNA microarray, that will tell the expression of a particular gene. So pharmacogenomics, the introduction, we said it is, I'm just going to give one example of a variation in individual which shows difference in the drug response. So one drug is called 6-mercaptopurine. This is a drug which is used for cancer. That is all means acute lymphoblastic leukemia. This is a childhood leukemia disease. For that, this mercaptopurine is the drug. Mercaptopurine is a drug because it will be equivalent to a DNA molecule. Say, for example, so this is a mercaptopurine structure, and this is equivalent to guanine. So here there is S instead of O, there is S, and then this H2N is not there. So this structure is similar to a nucleotide. So it is called a nucleotide analog. So how it helps in cancer? Because in cancer, uncontrolled cell division happens. Uncontrolled cell division is because of uncontrolled DNA replication. So we have to block the DNA replication. When we block the DNA replication, the cell division also can be arrested. So this is 6-mercaptopurine is a drug which is used as a drug. So whenever there is a DNA replication, this will go and bind to the growing chain, and then it will stop the replication. That is how this activity of this drug work. So this drug, sometimes it is some people react to that and some people they don't react. So thiopurine methyl transferase is the enzyme which will convert this where is that, which will thiopurine methyl transferase is the enzyme which is involved in converting inactivating this enzyme. 6-mercaptopurine will be converted to methyl mercaptopurine. So this will, TPMT is important for inactivating this enzyme. So active form is TGN. Once it is done, then it has to be inactivated, then it will be excreted. So some of the people, there are people have polymorphism, say TPMT activity. So this thiopurine methyl transferase activity is different for different individuals. Some individuals the activity is high and some individuals the activity is low. Depending on that, say for example, Caucasian population, 90% of the individuals have high TPMT activity. Only 10% have intermediate activity, 0.3 has low or no activity. So lack of TPMT activity results in the high risk of severe hematological toxicity. So although it is a medicine for cancer, if it is present in more amount in the system, it will lead to some toxic effect. So it has to be inactivated and thrown out of the cell. So some individuals it is thrown easily, some individuals it is not doing because this enzyme is impaired, because there are some mutations which accumulate in some individuals. So some wild type, the activity is high. When it is heterozygous mutant, the activity is low. So it is a variant, the activity is completely less. So if it is present in excess, it is harmful to the individual. So these are the different type of mutations which are present in that particular enzyme. That is TPMT is the enzyme and the gene corresponding to that and the mutations present in that. Some individuals, say you can see this, this particular individual, the activity is high. When the activity is high, it is good. It is going to degrade the mercaptopurine, because the other cells also have to divide and then make DNA and then divide. So TPMT genetic polymorphism in clinical consequence, low TPMT activity increased thiopurine toxicity. So some individuals, they have low TPMT activity because of the DNA polymorphism, they will have increased toxicity, increased risk of secondary neoplasm. So although it is a medicine for cancer, the secondary effect will be secondary neoplasm will happen because of the low TPMT activity. When high TPMT activity, decreased therapeutic effect. So what happens when it is, as soon as the drug enters in the cell, it is high TPMT activity means it is immediately inactivated. So there will be decreased therapeutic effect. So these are the various phenotype which is happening because of the polymorphism. So what they did after knowing all this, they improved the 6-mercaptopurine label. So this pharmacogenomic study ended in the improvement of the label. So in the label, earlier they said recommendation to use pharmacogenetic testing to guide dosing. So in order to decide on dosing, you have to use this label. You have to do pharmacogenetic testing. Like that is what given in the label of the drug. Now the new labeling is revised by sponsors in consultation with the FDA, includes data on increased risk of severe myelosuppression for TPMT activity deficient genotypes. So TPMT activity deficient genotypes will have some side effect of myelosuppression. So you be aware of that. So you have to always go with the genotyping. That is the end result of that. Similarly, there are so many other drug transporters and then polymorphism associated with several diseases like, so this transporter has strong evidence for cancer chemotherapy, weak evidence. These evidences are from different genotypes and then whether they have side effect because of the mutation. That is the evidence given here. Similarly, for drug target, this is guanine nucleotide G protein. So this is one target or a receptor and that variant and what are the polymorphism present in that drug. And then so the ultimate aim of this pharmacogenomic study is to have a targeted drug. Often drugs are only effective in specific subpopulation, responders, people who are responding to the drug. Early identification of responders can have a dramatic effect on treatment success. So if you are a responder, you can use the drug. Then there will be some cure for that. Treatment of non-responders puts these individuals at unnecessary risk. You are not a responder, but if you are taking that drug and that drug has lot of adverse events can cause in your body, and it is not a medicine, so it is like a toxin to you while providing no benefit. So you have to first find out whether you are a responder or a non-responder. Personalized medicine allows the identification of responders and non-responders for targeted therapies. This is happening today. This we call it as personalized medicine. So pharmacogenomics is genetic inheritance is only one factor in the drug response, but drug response phenotype, but understanding is increasing rapidly and the promise for enhanced drug safety and efficacy is real. So pharmacogenomics is happening. That means personalized medicine is there. So the vision of the pharmacogenomics is the right drug at the right dose for every patient. So genotyping an individual is important because each individual is different in their gene or genetic makeup, and that genes respond differently for particular drugs. So we have to genotype an individual before administering the drug to find out whether they are responders or non-responders. By doing that, we can avoid lot of adverse reactions for the drug. So that is the introduction for pharmacogenomics. So this is happening to date. Thank you.